Welcome to the video on Chapter 6 on enzymes. This is Dilly, my golden retriever. And the key topics in this video include learning about the physiological significance of enzymes, the classification of enzymes, how enzymes affect the rate of a reaction, the thermodynamic properties of enzyme-catalyzed reactions, the weak interactions between the enzyme and substrate, which are optimized in the transition state, and enzyme specificity and the diverse catalytic mechanisms that are employed. What are enzymes? Well, they are catalysts that increase the rate of the reaction without being used up. With the exception of a few catalytic classes of RNA molecules, they are all proteins. The study of enzymes dates back to the late 1700s. Life depends on powerful and specific catalysts known as enzymes, and almost every biochemical reaction is catalyzed by an enzyme, so they're extremely important. Enzymes vary greatly in size. Some require no chemical groups for their activity, but others require an additional chemical component called a cofactor, either one or more inorganic ions, or a complex organic or metallo-organic molecule called a coenzyme. This table shows you examples of inorganic ions that serve as cofactors for a variety of enzymes. This table gives you specific examples of coenzymes that serve as transient carriers of specific atoms or functional groups. For example, coenzyme A facilitates the transfer of acyl groups. Acyl groups, that's just a functional group that looks like this. That's an acyl group right here. The dietary precursor would be pantothenic acid. We also have lipoate, which transfers electrons and acyl groups. We have thymine pyrophosphate, which transfers aldehydes, and that is found in vitamin B1. So you'll actually notice quite a few of the B vitamins here that are the dietary precursors to the coenzymes. Many enzymes have been named by adding the suffix ase to the name of their substrate or to a word describing their activity. For example, urease catalyzes hydrolysis of urea, and DNA polymerase catalyzes the polymerization of nucleotides to form DNA. Other enzymes have been named by their discoverers for a broad function before a specific reaction catalyzed was known. The International Classification of Enzymes divides enzymes into 6 classes, each with subclasses based on the type of reaction catalyzed. Each enzyme is signed a 4-part classification number and a systematic name, which identifies the reaction it catalyzes. So in group 1, we have the oxidoreductases. They involve the transfer of electrons. In group 2, class 2, we have transferases that are involved in group transfer reactions. In group 3, class 3, we have hydrolases, which catalyze hydrolysis reactions. In group 4, we have lyases, which catalyze the cleavage of CC, CO, CN, and other bonds. In class 5, we have isomerases, which transfer groups within molecules to yield isomers. And then we have ligases, which form CC, CS, CO, and CN bonds. As an example of the enzyme classification, let's look at the enzyme that catalyzes this reaction, the conversion of ATP and D-glucose to ADP and D-glucose 6-phosphate. It is named D-hexo-6-phosphotransferase, and its EC number, enzyme commission number, is 2.7.1.1. The first number denotes the class name transferase. The second number denotes the subclass phosphotransferase, so what is being transferred. The third number indicates the group that's the acceptor, in particular the hydroxyl group. And the fourth number indicates that D-glucose is the phosphoryl group acceptor. This is a website where you can search for a particular EC number. You would just enter the number in the search there. and it gives you other names that are also associated with that particular enzyme. And then you can go up a level in the hierarchy, and then you see that you have phosphotransferases with an alcohol group as an acceptor. And there are a variety of those. And then if you go up to the subclass 2.7, you can see that now you have transferring phosphorus containing groups in that subclass. And then you can go up to the class of transferases, and you can see it gives a description. Transferases are enzymes transferring a group, for example, the methyl or glycosyl group, from one compound to another. Now, I would like you to go to the website and try using it. So I've assigned a particular EC enzyme to each of you. So find your name and go to that EC number and indicate in the chat what the enzyme is and give a description of the subclass. So for example, for Kenesha's, the subclass would be 4.3, the class would be 4. And notice that there is a class 7, and I told you that there were 6 classes. Well, actually, in August of 2018, the International Union of Biochemistry and Molecular Biology devised a new enzyme class called translocases. So pause the video, go to this website, find your particular assigned enzyme, and then give a description of the subclass in the chat.
I feel the need for speed. Ow!
Enzymes speed up reactions. Not all spontaneous reactions are fast. For example, consider the conversion of sucrose, known as table sugar, to carbon dioxide in water in the presence of oxygen. This is a highly exergonic process which releases the free energy that we use to think, move, taste, and see. However, a bag of sugar can remain on the shelf for years without any obvious conversion to carbon dioxide and water. How is that possible? Well, the reason is that although the chemical process is thermodynamically favorable, it is very slow. Yet when sucrose is consumed by a human, or almost any other organism, it releases its chemical energy in seconds, and the difference is catalysis. So without catalysis, chemical reactions such as sucrose oxidation could not occur on a useful timescale, and thus could not sustain life. This table gives you an example of some of the rate enhancements produced by enzymes. So carbonic anhydrase enhances the rate of those catalyzed reactions on an order of 10 to the 7th. That means that the reaction with the enzyme is 10 million times faster than the corresponding reaction without the enzyme. So you may be wondering, how do enzymes speed up reactions? The distinguishing feature of an enzyme-catalyzed reaction is that it takes place within the confines of a pocket on the enzyme called the active site. The molecule that is bound in the active site and acted upon by the enzyme is called the substrate. Now here's a question for you. In Chapter 5, what did we call a molecule that binds reversibly with a protein? Put your answer in the chat. This image shows the enzyme chymotrypsin with a bound substrate. Some key active site amino acid residues appear as a red splotch on the enzyme surface. Now we're going to talk about the thermodynamics of enzymatic catalysis. First of all, it's important to know that they do not affect the equilibrium, which means that they cannot affect the overall free energy of the reaction. What they can change is the activation energy for the reaction. So slow reactions face significant activation barriers, and enzymes speed up the reactions by decreasing this. And the delta G for the activation barrier is referred to as delta G with this symbol. Now let's look again at a reaction coordinate diagram that we've seen in the past. So here on the y-axis we have the free energy plotted, and then on the x-axis it's the progress of the reaction, so as the reaction proceeds. And then S would be the substrate, P would be the product. And this hill is the transition state. Now the energy between the substrate and the final product, that's the overall delta G for the reaction. So if that's negative, the reaction's spontaneous. If it's positive, it's non-spontaneous. This energy here, that's the activation energy. That's the energy required for the substrate to reach the high energy transition state. In order for the reaction to take place, the molecules, the substrate molecules, must overcome this hill, this energy barrier, and therefore must be raised to a higher energy level. At the top of the energy hill is a point at which decay to the substrate or the product is equally probable. It is downhill either way. This is called the transition state. The transition state is not a chemical species that can be isolated or has any significant stability, so it should not be confused with a reaction intermediate. It is simply a fleeting molecular moment in which events such as bond breakage, bond formation, and charge development have proceeded to the precise point at which decay to substrate or decay to product are equally likely. The difference between the energy levels of the ground state and the transition state is the activation energy. The higher the activation energy, the slower the reaction. The lower the activation energy, the faster the reaction. Think of the energy diagram like a roller coaster. The higher the hill, the slower the incline, and the greater the energy requirement to reach the top. Enzymes work by decreasing the activation energy. So here we have in the black line the uncatalyzed reaction, and in the blue line the catalyzed reaction. So we have two intermediates that are formed, the enzyme bound with the substrate and the enzyme bound with the product, called ES and EP. These intermediates occupy valleys in the reaction diagram. The overall activation energy is lowered in the catalyzed reaction versus the uncatalyzed reaction, which results in the reaction occurring much quicker. Now let's think about sucrose again. Here is the reaction written out for the oxidation of sucrose. Now this is a spontaneous reaction. It has a very large, very negative delta G. Yet sucrose is a stable compound because that activation energy barrier is quite high. So sucrose can be stored in a container almost indefinitely without reacting. However, in cells, it's readily broken down to carbon dioxide and water, and these enzymes not only accelerate the reactions, they also organize and control them so that much of the energy released is recovered in other chemical forms and made available to the cell for other tasks. The reaction pathway by which sucrose and other sugars is broken down is the primary energy-yielding pathway for cells, and the enzymes of this pathway allow the reaction sequence to proceed on a biologically useful timescale. The interaction between the substrate and the enzyme in the enzyme-substrate complex is mediated by the same types of non-covalent forces that stabilize protein structure. So we've got hydrogen bonding, ionic interactions, and the hydrophobic effect. And the energy derived from the enzyme-substrate interaction is called the binding energy. And its significance extends beyond a simple stabilization of the ES interaction. It's a major source of free energy used by enzymes to lower the activation energies of reactions. So much of the catalytic power of enzymes is ultimately derived from the free energy released in forming many weak bonds and interactions between an enzyme and a substrate. And this binding energy contributes to specificity as well as to catalysis. And weak interactions are optimized in the reaction transition state. Importantly, enzymes bind transition states best. This idea was proposed by Linus Pauling, and in this, enzyme active sites are proposed to be complementary to the transition state of the reaction, and they bind transition states better than the substrates. And stronger and additional interactions with the transition state as compared with the ground state lower the activation barrier. Now consider an imaginary reaction, the breaking of a metal stick. The uncatalyzed reaction is shown here in A. Now let's imagine two imaginary enzymes, two stickaces, if you will. We first design an enzyme perfectly complementary to the substrate shown in B. The active site of the stickase is a pocket lined with magnets. To react or break, the stick must reach the transition state of the reaction, but the stick fits so tightly in the active site that it cannot bend, because bending would eliminate some of the magnetic interactions between stick and enzyme. So look at the corresponding energy coordinate diagram. The enzyme substrate complex is so low in energy that it essentially forms this well, and then everything gets funneled there and can't overcome now this huge barrier to get to product. So such an enzyme would actually impede the reaction, stabilizing the substrate instead. So the ideal situation is where the enzyme is not complementary to the substrate, but to the transition state. So here, if we have an enzyme complementary to the transition state, the enzyme destabilizes the stick, contributing to catalysis, and the binding energy of the interactions compensates for the increase in free energy required to bend the stick, shown here. Now you see in this reaction coordinate diagram that the activation energy is lowered, but the ES is not so stable as to impede the reaction from progressing. Now that was an imaginary reaction, but real enzymes work on an analogous principle. So some weak interactions are formed in the enzyme-substrate complex, but the full component of such interactions between substrate and enzyme is formed only when the substrate reaches the transition state. The free energy or binding energy released by the formation of these interactions partially offsets the energy required to reach the top of that energy hill. Even on the enzyme, the transition state is not a stable species, but a brief point in time that the substrate spins atop an energy hill. The enzyme-catalyzed reaction is much faster than the uncatalyzed process, however, because the hill is much smaller. The important principle is that weak binding interactions between the enzyme and a substrate provide a substantial driving force for enzymatic catalysis. The groups on the substrate that are involved in these weak interactions can be at some distance from the bonds that are broken or changed, and those weak interactions formed only in the transition state are those that make the primary contribution to catalysis. Conceptually, specificity is easy to distinguish from catalysis. However, experimentally, this distinction is often more difficult to make because they both arise from the same phenomenon. So specificity is referring to an enzyme specifically binding particular substrates. So it's able to discriminate between a substrate and a competing molecule. If an enzyme active site has functional groups arranged optimally to form a variety of weak interactions with a particular substrate in the transition state, the enzyme will not be able to interact to the same degree with any other molecule, hence giving it specificity. For example, if the substrate has a hydroxyl group that forms a hydrogen bond with a specific glutamate residue on the enzyme, any molecule lacking that hydroxyl group at that particular position will be a poorer substrate for the enzyme. So let's discuss more about how enzymes lower the activation energy. First, the binding energy constrains the substrates in the proper orientation to react. So all reactions require a collision of molecules, and they must collide at just the right orientation. And those types of collisions do not occur randomly in solution that often. Indeed, they can be exceedingly rare. Substrates can be precisely aligned on the enzyme, however, with many weak interactions between each substrate and strategically located groups on the enzyme clamping the substrate molecules into the proper positions. Second, formation of weak bonds between substrate and enzyme results in desolvation of the substrate, and the enzyme-substrate interactions replace most or all of the hydrogen bonds between the substrate and water that would otherwise impede the reactions. Here is a picture of that happening. Third, binding energy involving weak interactions formed only in the reaction transition state helps to compensate thermodynamically for the unfavorable free energy change associated with any distortion, primarily electron redistribution that the substrate must undergo to react. And fourth, the enzyme itself will undergo a conformational change when the substrate binds, and this is referred to as induced fit. Enzymes may use one or a combination of the following: acid-base catalysis, where the enzyme gives and takes protons; covalent catalysis, that can involve the changing of reaction pathways; and metal ion catalysis, using redox cofactors. So here we see how a catalyst circumvents unfavorable charge development during cleavage of an amide. The hydrolysis of an amide bond shown here is the same reaction as that catalyzed by chymotrypsin and other proteases. Charge development is unfavorable and can be circumvented by donation of a proton by H3O+, or HA, where HA represents any acid. Similarly, charge can be neutralized by proton abstraction by hydroxide or any general base. Many organic reactions that are used to model biochemical processes are promoted by proton donors or proton acceptors. The active sites of some enzymes contain amino acid functional groups, such as those shown here, that can participate in the catalytic process as proton donors or proton acceptors. So these would be examples of your proton donors, and these would be examples of your proton acceptors. A transient covalent bond can also form between the enzyme and the substrate, and that will change the reaction pathway. So for example, if the uncatalyzed pathway involved cleavage of the AB bond with water, the catalyzed pathway involving catalyst X would go through this pathway instead. So notice that in the catalyzed pathway, you're forming a bond between A and the catalyst before you get to the final products of A and B separate, and then the catalyst has been released as well. These types of reactions require a nucleophile on the enzyme. Finally, metal ion catalysis involves a metal ion bound to the enzyme, and it can interact with the substrate to facilitate binding or stabilize negative charges and can participate in oxidation reactions. Now I have a few questions here to test your understanding. So put your answer in the chat and give your reasoning. So here's a reaction. What type of enzyme do you think catalyzes this reaction? An isomerase, a hydrolase, a transferase, a ligase, an oxidoreductase, or lyase? Pause the video and put in your answer in the comments. Next question, how does an enzyme speed up a reaction? Look over the options and put your answer in the chat. We've discussed in this video the conversion of sucrose to CO2 in water, and that it has a very large negative delta G. So why does the conversion not happen readily in your kitchen pantry, but it does happen quickly in your body? Explain this using thermodynamic parameters and put your answer in the chat. If an enzyme accepts H+ from hydronium and transfers it to an amine group of the substrate, this can result in an increase in the rate of release of the product. What would this be an example of? Induced fit, acid catalysis, covalent catalysis, or entropy reduction? Put your answer in the chat. Finally, how does the induced fit mechanism of enzyme catalysis work? A, the enzyme assumes a conformation identical to the substrate. B, the enzyme undergoes a conformational change to maximize weak interactions to the substrate. Or C, the substrate changes its conformation to fit the enzyme. Again, put your answer in the chat. Congratulations, you finished this video. I'll see you in class.